Well, thank you very much to the organising committee for inviting me to talk. I think hopefully it's going to be a very interesting session. I'm very much looking forward to hearing about the things that I don't do, which are much newer than the things that I'm going to talk about now in the present talk. So I'm going to give an introduction to calibrated fMRI approaches for mapping oxygen extraction fraction particularly, but our interest in this started a few years ago when we wanted to look at the amount of oxygen that the brain is consuming. I start off with acknowledgements for the people who funded this work. More recently, support from the Italian government, but for many years before that in the UK, and particularly to Mike Gamushka, who really is, for the last number of years, has been the brains behind a lot of the developments that we've done. And I'm trying to convince the team working with me now in Italy to take this forward as well. So why do we want to map oxygen extraction fraction? Well, it's one of the important physiological parameters that will help us understand brain tissue function. So it can give us an idea of whether the brain's blood vessels are working properly. And it's one of the crucial parameters that will help us calculate tissue energy consumption, or rather oxygen, the rate of oxygen metabolism. And in doing those things, we get a better understanding of disease mechanisms. We might be able to assess treatment interventions in the future to find out whether they're working. And, you know, there's a question mark about whether this might eventually become a clinical tool. And I'm sure we'll have some discussion on that later on. Well, oxygen extraction fraction, as a fairly long literature will tell us, is sensitive to compromised cerebral blood flow. So this slide here is showing data from PET studies. This is oxygen 15 PET. And you can see that where there's an area of reduced cerebral blood flow here, there's an increase in oxygen extraction fraction. So the proportion of oxygen that's being extracted from the blood goes up to try and compensate for reductions in blood flow and maintain oxygen delivery to brain tissue. And oxygen delivery is described in terms of an oxygen consumption is described in terms of this mass balance expression, where we're looking at the product of the concentration of oxygen in arterial blood, the fraction of oxygen that's being removed from the blood as it goes through the brain times blood flow. So in this case, oxygen extraction fraction is telling us that the blood flow is probably not sufficient in the areas where it's reduced, or at least it's been heavily reduced. Now, as I mentioned, we're interested in quantifying oxygen extraction fraction because it's necessary to calculate brain oxygen consumption. And this, albeit simplified but still complicated diagram, is just to remind us that most of the energy that enters the short-term storage in the brain as ATP is released oxidatively. Of course, to get a full picture of energy consumption, we also want to measure glucose consumption, but oxygen in most brain tissue, oxygen usage is a good measure of how much energy is being consumed. So this is the typical physicist's eye view of the brain. As I've explained, oxygen consumption is the product of these three quantities, but our main challenge here is to measure venous oxygen content, and that's what gives us oxygen extraction fraction. We can measure the concentration of oxygen in arterial blood fairly easily either through pulse oximetry measurements or direct blood gas measurements, or even inferring it from measurements of expired oxygen from the lungs. Blood flow, we have a number of ways of measuring blood flow using MRI, as you know, one of them mapping techniques being arterial spin labeling, but oxygen extraction fraction is the challenging one which we're focusing on today. So this is about measuring the we want to know the concentration, really, of venous deoxyhemoglobin. Now we're going to hear about some of the other techniques after my talk, so I won't focus on these, but broadly, we can think of that there are a number of different measurement methods. So we're talking today specifically about mapping. So we want to try and get some spatial discrimination of where of our oxygen extraction fraction measurements. But probably most important in the literature so far have been the whole brain or regional approaches, particularly TRUST, T2 based methods looking at large blood vessels. The robustness of those signals is very good, but these are intravascular. These are based on intravascular measurements in big blood vessels, which gives us relatively poor spatial resolution, or rather gives us a global measurement of what's going on across the brain. Today we're focusing more on mapping. And again, you could think about the techniques being divided into those that try to look at an intravascular signal based on the oxygenation of blood within the vessels, and other techniques, for example, T2 mapping, and other techniques which rely on changes in susceptibility of the blood, which have an effect outside the small blood vessels as well as inside them. So those might include the QBOLD and QSM type approaches, as well as what I'm going to talk about, which is based on the good old BOLD effect of almost 30 years ago, which has developed into this so-called calibrated fMRI, or we often call it dual calibrated because we're calibrating using two gases, or QO2 or QO2 from which has been coined by Claudine Gauthier from the original work almost 10 years ago now. So what are we trying to do? Well, more than 20 years ago, the Davis model was proposed, this sort of phenomenological model to describe changes in BOLD signal here, this delta S, this fractional change in BOLD signal in terms of a maximum BOLD signal change dependent on local blood volume and deoxyhemoglobin concentration. And this calibrated approach to measure absolute oxygen consumption and oxygen extraction fraction is really just a development of that approach. Now, in the original work of Tim Davis and then Rick Hogue, it's possible to measure relative changes in oxygen consumption with a single calibration step using hypercapnia, which increases blood flow. So as carbon dioxide levels increase in arterial blood, you see an increase in blood flow and an increase in BOLD signal up to a maximum when you lose all of the deoxyhemoglobin. So that's one calibration step. The second calibration step, you need to separate the effects in the BOLD signal of blood volume and oxygen extraction fraction. Both of those have an effect on the BOLD signal. So we need a second calibration data point, if you like, to be able to separate those things. So the second thing is to introduce hyperoxia, and this increases arterial concentration of oxygen, and thereby also increases the concentration of venous oxygen, on the assumption that the brain's oxygen metabolism doesn't change when you do these interventions. So both of these, both the hypercapnia and the hyperoxia, assume isometabolism, which is one of the physiological assumptions that we make, which is debatable. There's some work suggesting that you get changes in oxygen metabolism with CO2 administration. But essentially, we've got this mass balance expression down here where we equate baseline oxygen consumption with oxygen consumption during changes in carbon dioxide and oxygen levels. And if we put these together, we get an expression that looks unpleasant, but it's relatively straightforward. It's got the things that we want to know, the concentration of deoxyhemoglobin. We can measure the concentration of arterial oxygen. We measure changes in BOLD signal, and we measure changes in blood flow using arterial spin labeling techniques. So then it's a question of designing an experiment to modify oxygen levels, modify carbon dioxide levels, and have an appropriate MRI measurement technique to acquire all the signals. We worked for a number of years preceding this work on controlling carbon dioxide levels and oxygen levels inside the MRI scanner. And we've used a number of different systems of oxygen and CO2 control, but the aim really here is to get some step changes in carbon dioxide through an experiment here, which lasts about 20 minutes, and step changes in oxygen to produce these two different modulations. You can see here examples of end tidal oxygen levels shown on the left and carbon dioxide changes shown on the right. We take our end tidal values as representative of changes in arterial concentrations. So what we expect to see down on the bottom right here are changes in the bold signal, or T2 star weighted signal, that arise from increases in blood flow and carbon dioxide and increases in oxygen. But the increases in oxygen don't really produce a change in cerebral blood flow. So we have discrimination, if you like, of the effects of carbon dioxide and oxygen in the bold signal and in an arterial spin labeling blood flow signal. We've used, most recently, we've used an acquisition pulse sequence that combines arterial spin labeling with bold, but in separate volumes. So we don't use a multi-echo approach, and this, as we did originally. So our first work used a multi-echo arterial spin labeling sequence, but we've since moved to a sequence which acquires bold signals straight after the ASL volumes. The reason for that is that it allows us to do background suppression of the ASL without losing so much bold signal, given that we're largely limited by signal to noise in the ASL acquisition. So it improves the ASL data, as most of you know, to do the background suppression. And so you can see an example of a blood flow signal on the top left and bold signal variation on the top right here. And Mike Ganushka has published a series of papers where we've gradually improved the data fitting or parameter estimation methods. So starting off with a regularized nonlinear least squares a few years ago to produce, you can see example maps of single subject here, oxygen extraction fraction, blood flow at the bottom, and oxygen consumption in the middle. Mike has since improved that with a machine learning approach. Address questions to Mike, as I'm not an expert in those techniques at all, but essentially you can see that what we get if I toggle between the two is we get flatter OEF maps. So this is the original data estimation technique, and then with the machine learning we see a flatter oxygen extraction profile, which is, you know, it makes us think this is doing slightly better, because PET methods seem to tell us that oxygen extraction is pretty flat across healthy cortex, at least. I should say that we only really trust the measurements that we've got here in grey matter, because as you know, arterial spin labeling is weak in white matter because of the long arrival times. So this machine learning approach seems to do a bit better with the noise in the data, giving us slightly more confidence in our estimates. And we can also see this when we look at correlation between oxygen extraction fraction and hemoglobin concentration that we measure in our participants. This is important for getting a good estimate of oxygen consumption. We need to know how much oxygen the blood is carrying. So if we look at the machine learning approach down the bottom, we see a tighter correlation between hemoglobin concentration and oxygen extraction fraction, which is what we would expect. Now, validation of these methods is, you know, is always tough. None of us really wants to go and do a PET study for various reasons. Audrey Fan and Claudine Gauthier published a nice study a few years ago where they did a validation at 70, so a comparison of gas calibrated approaches with a susceptibility based approach to oxygen extraction fraction measurements. So we have a clear correlation here between the two techniques. One of the problems with validating oxygen extraction fraction is that there's a pretty narrow range of oxygen extraction fraction in healthy brain anyway. And so, you know, we don't have a lot of underlying true variability to validate against. We've got some data unpublished, which we're working on at the moment, which is a similar principle. So this is at 3 Tesla, and we obtained a very similar correlation between our calibrated approach on the x-axis here, and then a phase-based method. This is the Oxflow method from Felix Verley's group. So ultimately, we can, you know, we might call this measurement from this single scan, which lasts about 20 minutes, unfortunately. It's a long scan, a kind of toolbox offering us measurements of blood flow, vascular reactivity, oxygen extraction fraction, and oxygen consumption. I mentioned vascular reactivity. One of the positive things, if you like, of going to all the trouble of doing these gas challenges is that you get other measurements besides oxygen extraction fraction. So vascular reactivity, as you know, as previous talks in this group have concentrated on, gives us interesting information about vascular function. Just in the last few minutes of my last 5 minutes of this introduction, I just want to show you some examples of where these calibration techniques have been used so far. And I guess there's a kind of evolving, you know, there's an evolving set of studies, not a huge number, but last 5 years or so, people have started trying to use these techniques. This is unpublished data in a single patient. This is a patient who was due to undergo surgery for epilepsy, and we can see on a PET scan an area of reduced glucose consumption based on FTG in the temporal lobe. We got the patient to do our CMRO2 mapping procedure. So I'm not showing OEF here, I'm showing the oxygen consumption, and you can see with the eye of faith at least that we appear to have a reduced oxygen consumption within the same region of brain. This was also reflected in the arterial spin labeling blood flow map. So oxygen extraction fraction probably wasn't that impaired, but the overall oxygen consumption was in this patient. So this is kind of anecdotal single patient evidence. Jill Davis has published 2015, I think, a nice study where she looked at a decent cohort of young and elderly participants using these calibrated methods in order to characterize differences in blood flow, oxygen extraction fraction, which appeared to be slightly reduced, and oxygen consumption, baseline oxygen consumption in healthy aging, as you can see here. We've looked at pharmacological effects. So we published a study on caffeine, looking at the effects on oxygen consumption and oxygen extraction fraction. We expect caffeine to have effects directly on neuronal activity, but also we know that caffeine has vasoconstrictive effects. So we anticipate that with a change in blood flow, we might, if we're preserving oxygen consumption, we'd expect to see perhaps an increase in oxygen extraction fraction. So what do we see that? Well, you can see here in a caffeine administration session versus placebo, we have a reduction of about 30% in this case of blood flow. The oxygen extraction fraction appears to go up by close to 20%. So it might go up from 0.4 to 0.48 or 0.45, something like that, which doesn't quite compensate, in this study, doesn't quite compensate for the drop in blood flow. So we do see an overall reduction in oxygen consumption. Now this was a mapping study. So we produced maps of with statistical thresholds of oxygen extraction fraction, which you can see increasing. There's as we know with caffeine, there's a very widespread reduction in blood flow. Applying a statistical threshold, we get a kind of patchy reduction in CMRO2, but you can see areas of strongly increased oxygen extraction fraction, where changes in CMRO2 are minimized. So it's possible, we've shown, here in healthy volunteers, a modulation of oxygen extraction fraction with a pharmacological intervention. I should say caffeine's you know, there's kind of not controversial. There are varying results with caffeine about its effect on CMRO2, but I think the effect on oxygen extraction fraction is fairly robust. If blood flow goes down, we expect OEF to go up if the brain is still using a similar amount of oxygen. We've extended recently to a cohort of multiple sclerosis patients where we hypothesize that inflammation is compromising tissue oxygen usage. This is relapsing remitting MS. This is study's not yet published. You can read it on bioRxiv, but it's under review in a journal at the moment. Looking at a single patient, this is a single MS patient on the top row and a single control on the bottom row. The MS patient CMRO2 looks more patchy. If we go and look at whole brain blood flow, oxygen extraction fraction, and oxygen consumption, blood flow is reduced significantly in patients. Oxygen consumption significantly reduced, but oxygen extraction fraction is not, which suggests, could suggest a number of things, but it suggests that perhaps one of the interpretations is that the MS brain is not demanding as much oxygen. There's an impairment in the tissue, which means that it's not trying to use as much oxygen. So the oxygen extraction fraction stays, you know, oxygen and blood flow remain coupled and oxygen extraction fraction remains similar. That's not entirely consistent with studies of, with the whole brain studies based on T2 measurement, where I think a difference in oxygen extraction fraction was found. But we do notice that when we look regionally across grey matter, we see that this reduction in CMRO2 seems pretty global. So the unthresholded and thresholded results in the two different rows here. Finally, just to show that, you know, a number of other groups are doing this as well. 3 years ago now, or sorry, 4 years ago, Rick Hoag's group published a study in Alzheimer's disease looking at oxygen consumption. So we've got blood flow on the top row, oxygen extraction fraction, which is reduced in Alzheimer's disease here, and then essentially multiplying the effect with reduced blood flow, we see a bigger, they saw a bigger reduction in oxygen consumption. So the oxygen consumption changes appear to be compounded by changes in blood flow in Alzheimer's disease. So there's pros and cons with all of these methods. Calibrated fMRI approaches have been used now for a number of years. We and others have demonstrated some success for them in mapping oxygen extraction fraction, but you know they do rely on respiratory challenges, which are more trouble to do experimentally, and they rely on arterial spin labeling blood flow measurements, measuring changes in blood flow, which have limited signal to noise, and so we do have problems in white matter. And also, we would be likely to have problems in clinical conditions where blood flow is heavily reduced. And I expect we can discuss some of these things a bit later on. Thanks very much for listening.
Thank you. Thanks.